Welcome back to the Deep Dive. Today, we're stepping away from maybe the more familiar threats, anthrax, smallpox, even things like weaponized Ebola.
Right.
We're going into a really specialized and honestly pretty unnerving area. We're talking about biological threats designed not for that big, obvious pandemic, but specifically for clinical and diagnostic evasion.
Exactly. We're looking at bioweapons engineered not just to harm, but potentially to completely defeat medical attribution, diagnosis, even pathology.
That's the core of it.
Yeah, and our source material today, it draws a lot from those 1997 Jason group assessments, also analyses of synthetic biology advances, and importantly, historical accounts from offensive programs, particularly the Soviet efforts.
Okay.
And these sources, they really challenge some fundamental assumptions in medicine. So for you listening, if you're a specialist physician, an ID expert, a pathologist, maybe an oncologist, our mission here is to unpack the concept of stealth bioweapons.
Stealth bioweapons.
These are agents, often genetically tweaked, designed for cryptic effects, maybe delayed onset, or engineered to perfectly mimic natural diseases. The key problem is making the line between, say, an endemic surge and a covert attack, well, diagnostically invisible.
The stakes just feel incredibly high here. For the clinician, it shifts the challenge, doesn't it? It's not just treating a known bug anymore.
Not at all.
It's trying to figure out if a patient's really severe, maybe idiopathic autoimmune condition, or I don't know, an unusual cancer cluster that pops up, is that actually the result of some hidden engineered intervention?
That's the nightmare scenario.
So the central question for this whole deep dive seems pretty simple, but also terrifying. What happens when an illness looks completely natural, indistinguishable from the background noise of disease we see every day, but it absolutely isn't?
Right, and that forces us to look way beyond just traditional infectious disease tracking.
Yeah, it pushes into genomics, long-term pathology, chronic disease surveillance.
Yeah.
It's a much bigger picture.
It really forces the medical community to grapple with the idea that the technological line between a bad natural outbreak and a sophisticated covert attack is becoming, well, diagnostically blurry, opaque even.
Okay, so let's start unpacking that. Where do we begin? The blueprints for evasion, maybe.
That's the place. Let's start with the theory.
All right, the blueprint of evasion. You mentioned the Jason group. Let's start there with this idea they flagged the stealth virus.
Okay, so the Jason group, basically it's this elite group of academic scientists the government tasks with looking over the horizon at future threats. Back in the late 90s, they reviewed biotech progress.
And they saw potential problems.
Big ones. They identified about 6 broad groups of potential future engineered pathogens, and the stealth virus concept was a major point of concern for them.
So for our audience, the clinicians listening, how should we define that? That sounds like more than just a virus that's tricky to culture in the lab.
Oh, absolutely. Way more insidious. A stealth virus, as conceptualized, is really designed for cryptic infection. The goal is to clandestinely infect and then persist within the genome of a target population.
Persist in the genome. Okay.
Yeah, its whole signature is long-term, undetectable latency. It avoids that fast, acute presentation you associate with traditional bioweapons. The engineering goal here is really genomic integration and just hanging out, undetected.
That idea, genomic persistence, and then a delayed clinical effect, that throws a massive wrench into standard public health epidemiology, doesn't it?
Completely.
I mean, if the symptoms only show up years, maybe even decades later, your whole standard timeline tracing exposure, finding the source, it's gone. How do you ever attribute the sickness or death back to an event?
And that's precisely the core strategic value of stealth. It breaks the chain of attribution. Let's detail some potential functions they envision. First, delayed activation. The virus is engineered to just lie dormant. Maybe it integrates into host DNA, like some retroviruses do, and only becomes clinically active much, much later in the people targeted.
So the link between exposure and pathology is just severed by time.
Exactly. By the time a physician actually sees the patient with symptoms, the initial attack, the exposure event, is ancient history. The trail is ice cold before the first symptom even shows up.
And stemming from that delay, there's another potential use often discussed in these threat assessments, blackmail potential.
Right. The threat itself becomes the weapon, just the knowledge or the claim that a certain population segment carries this genetic time bomb that could be triggered remotely or on a timer.
That could be used as a political lever, a coercive tool, even if the weapon itself is never actually activated.
The geopolitical leverage comes purely from the uncertainty embedded in the genome. But perhaps the most critical function, especially for oncologists, tumor boards, chronic disease specialists listening,
Yes.
is the third possibility, inducing cancer.
How would that work?
Well, the source material suggests a stealth virus could be engineered to carry DNA segments that mimic or are actually just activated oncogenes.
Oncogenes, the genes that kick off unregulated cell growth, basically.
Right. Those specific DNA segments known to initiate cancer. So you're talking about an engineered mechanism to deliberately promote malignancy. It hijacks the cell's own regulatory machinery.
Kind of like how we know high-risk HPV or certain retroviruses can work, but delivered covertly, deliberately.
Precisely. These engineered genetic segments could, directly or indirectly, cause various cancers. But the crucial point is the timeline.
The delay again.
Exactly. The clinical effects, solid tumors, chronic leukemias, might take 5, 10, maybe 20 years to fully manifest. And critically, from an offensive program's perspective, this slow timeline is seen as a viable strategic option.
Because it maximizes deniability.
Absolutely. It fatally complicates attribution. Think about it. A sudden anthrax outbreak. That's traceable to a date, maybe a place. But an unexplained, persistent, say, 15% rise in a specific lung cancer type in a certain demographic, appearing 5 or 10 years after some potential unknown event.
That just gets lost in the noise.
It becomes statistically indistinguishable from background rates or maybe environmental factors, other confounders. It's deniable.
Wow. That really does shift the problem entirely away from just acute infectious disease response. Now you're talking about needing long-term chronic disease surveillance infrastructure to even pick up the signal.
Correct. And it poses huge challenges for genomic sequencing and pathology, too.
How so?
Well, think about it. If a viral fragment integrates into the host genome and then triggers cancer down the line, how does a standard tumor board tell that engineered exogenous trigger apart from just a spontaneous mutation caused by, say, smoking or random chance?
Standard tumor sequencing panels, they're looking for known common mutations, right? Or known activated oncogenes.
Exactly. They aren't typically designed to scan the entire genome looking for rare, highly divergent, or secretly integrated external viral sequences. You'd need really deep, non-standard genomic sequencing, specifically looking at integration sites, promoter activity, things like that. Only then could you even begin to rule out an engineered trigger.
So we're essentially asking the specialists who treat the consequence of tumor to also be the first line of defense against a potential act of aggression. They become forensic geneticists by necessity.
In a way, yes. It's a profound shift.
Okay, that sets things up perfectly.
Yeah.
Let's move from this genetic theory of latency into the more immediate practical ways these things might be engineered to defeat our labs, our ERs, right now.
Right, because standard biological weapons already have some stealth, you know? It's inherently hard sometimes to tell a deliberately released flu strain from a nasty natural one, especially early on.
Sure.
But modern biotech just gives attackers so many more tools to enhance that concealment. We're moving beyond just general latency now into specific targeted diagnostic sabotage.
Okay, let's focus on that first major mechanism, evasion of detection and treatment.
This is the functional nightmare for anyone running a clinical lab, molecular diagnostics people, ID physicians. Pathogens can be genetically manipulated to be harder to detect, or, even worse, specifically designed to defeat the identification methods we rely on right now.
Like PCR.
Exactly like PCR. Think about engineering a pathogen to have a slightly different, maybe highly divergent genetic sequence right at the key binding sites, the spots where our common diagnostic tools, like PCR primers, are designed to latch on.
So our whole diagnostic world leans heavily on techniques like PCR, which targets those highly conserved genetic regions, the bits that are usually stable within a species or genus.
Right.
If an attacker strategically mutates just those short sequences where the primers need to anneal, maybe without even affecting the bug's function otherwise.
Bingo. If the agent is designed to bypass standard PCR or maybe antibody assays too by introducing these strategic non-functional changes right at the primer binding site, maybe in the 16S RNA gene region for bacteria, for example,
The result is a false negative.
a guaranteed false negative on our fastest, most common tests. And that technical evasion, it means the time lag until you get definitive attribution, which now requires full genomic sequencing, comparing it to databases, gets stretched out dramatically.
During that delay.
During that delay, the event gets misclassified, it's a natural occurrence, maybe a misdiagnosis, effective quarantine is delayed, the right treatment is delayed, chaos ensues, potentially.
And this capability, this evasion, it's not just about detection, is it? It goes into treatment, too.
Oh, absolutely. There's historical context here. The former Soviet Union's bioweapons program, sources tell us, specifically prioritized agents for which there was, and this is the key phrase, no prevention and no cure.
No prevention and no cure.
Now, apply that concept with modern genetic engineering. It's terrifying. You can engineer a pathogen with multiple layered drug resistance genes before it's even released. Resistance to macrolides, fluoroquinolones, cephalosporins, you name it, all designed in from the start.
So the modern version of no cure is just making it resistant to everything in the pharmacy.
Pretty much. You create a bug that's genetically resistant to every available antimicrobial class. It renders our entire infectious disease toolkit obsolete on arrival. The clinician is left treating a highly virulent, highly resistant organism with effectively zero therapeutic options.
That is sobering. And it leads us, I think, to the most insidious level of stealth, where the goal isn't just to fool the lab, but to fool the pathologist, the medical examiner.
Right.
We're talking about what you call the ultimate attribution nightmare, where advanced research aimed to leave literally no trace of outside intervention.
Yeah.
Making the ME the last and maybe most challenged line of defense.
Absolutely. When the absolute highest strategic priority is attribution failure, making sure no one can ever prove you did it, then offensive programs turn inward. They look for ways to leverage the body's own systems for destruction.
Okay, let's unpack that. Mechanism 2A you mentioned, triggering autoimmunity.
Yeah, researchers like Sergei Popov, who later defected from the Soviet program, they talked about seeking methods to trigger a severe autoimmune response. Instead of using classic pathogens or big foreign toxins, the idea was subtler.
Oh, what's the mechanism?
It often involves something called molecular mimicry. You engineer an agent, maybe a virus, maybe something else, to introduce a protein that looks structurally very similar to one of the host's own natural proteins, a self-antigen.
Like a protein found in kidney tissue or joints or heart muscle.
Exactly. Something critical. Then the immune system does its job. It mounts a response against this foreign engineered look-alike protein.
But because it looks so much like the body's own protein, the immune response cross-reacts. It starts attacking the host's own tissues.
Precisely. So what does the physician see? They see conditions that look clinically identical to known, severe, often idiopathic autoimmune disorders, maybe an acute onset lupus or a really aggressive rheumatoid arthritis, necrotizing vasculitis.
Or even something like sudden overwhelming anaphylactic shock or sudden cardiac death from inflammatory myocarditis.
Potentially, yes. So for rheumatologists, ER docs, internal medicine specialists, this is the ultimate diagnostic puzzle. The actual damage, the pathology, it's entirely endogenous. The body is attacking itself. There's nothing obviously external, no weird bacteria, no exotic toxin defined.
So how do you distinguish a natural, really bad autoimmune flare-up from one that was deliberately engineered through this subtle molecular mimicry?
It's incredibly difficult. You'd probably need to look for very specific, maybe rare autoantibodies or atypical patterns in the serology tests, or maybe you'd notice an unusual clustering of these severe cases in a population that doesn't normally have the genetic predisposition for these kinds of diseases. It requires a very high index of suspicion.
A staggering challenge. Okay, if that level of engineered self-destruction wasn't enough, let's move to mechanism 2B, using untraceable, naturally occurring substances, regulatory peptides.
This might represent the absolute peak of forensic stealth. We're moving beyond infectious agents entirely here. Now we're talking about biological weapons based on chemical substances the human body produces naturally. Specifically, very potent regulatory peptides.
These are the body's own signaling molecules, right?
Mm-hmm.
Hormones, neuropeptides, growth factors, things that control everything.
Exactly. They govern internal functions, mood, nerve function, heart rhythm, metabolism, you name it. They're essential.
So weaponizing them, what kind of effects could that have?
Well, it depends entirely on which peptide you target, but the effects could be broad and devastating. You could potentially damage the nervous system, cause drastic mood alterations, trigger severe psychological changes, maybe induce rapid, overwhelming cardiovascular collapse.
This brings neurologists, psychiatrists, cardiologists directly into the biodefense equation.
It does. But the critical factor here, the reason this is the ultimate attribution nightmare, is the failure at the pathology stage. Why were these endogenous peptides so interesting to people designing covert weapons? Because of how quickly they're metabolized. And because a sudden spike can look identical to a natural catastrophic event, Our sources, particularly those discussing historical research, possibly linked to the KGB, suggest regulatory peptides were prioritized specifically because they could not be traced by pathologists.
Can be traced.
Many of these peptides have incredibly short half-lives in the body minutes, sometimes even seconds. So by the time a victim is discovered, by the time an autopsy can be performed hours or days later, the engineered spike of that external peptide is long gone, metabolized, vanished.
So the medical examiner finds the effect, maybe massive heart damage, a brain hemorrhage, signs of a cytokine storm.
Exactly. They see the result of the catastrophic failure. They might even find high levels of the body's own stress hormones or other signaling peptides, but that's just the natural consequence of the body dying catastrophically, like from a massive heart attack or stroke. They can't distinguish the engineered external trigger from a purely natural internal malfunction.
So the victim appears, clinically, diagnostically, and pathologically, to have died of natural causes.
Perfectly natural causes. The external intervention is effectively erased post-mortem by the body's own metabolic cleanup crew.
This isn't just theory, then. This moves us squarely into a specific historical example. Section 4. The myelin toxin developed under that Soviet program called Bonfire. This seems to illustrate the peptide and autoimmune strategy in terrifying detail.
Right. The Bonfire program was reportedly intensely focused on developing these kinds of regulatory peptide and protein weapons. They were relentlessly searching for that perfect, untraceable killer that would just sail past any standard pathological review.
Okay, so let's detail this myelin toxin for our listeners, especially those in neurology. What was the specific mechanism they were trying to engineer?
It was, frankly, a brilliant piece of malicious genetic engineering. Reports suggest scientists in the program identified genes that coded for small but highly immunogenic protein fragments of myelin.
Myelin, the fatty insulation around our nerve fibers, absolutely critical.
Critical. They took these myelin fragment genes and inserted them into some kind of viral vector, likely a relatively harmless virus that could efficiently infect people without causing much fuss on its own.
So the virus itself isn't the main weapon here. It's acting like a delivery system, like a silent, targeted gene therapy tool.
Exactly. Its sole purpose is to deliver the genetic code that then tricks the body's immune system into attacking the nervous system.
How?
Once the viral vector gets that myelin protein fragment gene into the host cells, those cells start producing these fragments. They're seen as foreign by the immune system, which mounts a powerful attack against them.
But because those fragments are designed to perfectly mimic the real myelin sheath covering the person's own nerves.
The immune system cross-reacts. It can't tell the difference. It starts systematically destroying the body's own vital nerve insulation in the brain, the spinal cord, peripheral nerves.
The clinical outcome would be devastating. Massive demyelinating disease, something far worse than normally seen.
The spectrum could potentially mimic something like acute disseminated encephalomyelitis, ADEM, or maybe a super-aggressive, rapidly progressive form of Guillain-Barré syndrome. It could lead to widespread paralysis, neurological collapse, and death, perhaps within days or weeks.
But crucially, it would likely be recorded as what? An idiopathic demyelinating disorder. Severe neurological failure of unknown cause.
Precisely. For a neurologist seeing a cluster of unusually severe, maybe atypical, unexplained cases like this, especially if it's happening in a population without any preceding viral illness or known genetic risk factors, the possibility of an external engineered autoimmune trigger has to become part of the differential diagnosis. A chilling thought, but necessary.
It really highlights that chilling link between, basic immunology research concepts like molecular mimicry and actual operational strategic weapon development. But let me ask this. If predicting the exact outcome is still technically difficult with these complex genetic systems, wouldn't something simpler, like a fast-acting conventional neurotoxin or poison, still be preferred for, say, a targeted assassination? Are we maybe overstating the current operational readiness of GWS?
That's a really crucial question, and the sources do touch on this trade-off. Conventional poisons, yes, they offer precision. You know the timing, you know the dose response pretty well, but they fail that ultimate attribution challenge, the pathology review.
Because they leave traces.
Exactly. Conventional toxins leave metabolic markers, maybe heavy metals, specific degradation products. A good modern forensics lab can eventually find those traces. Genetic weapon systems, or GWS, they essentially trade that immediate tactical precision for long-term strategic deniability.
So if the primary goal isn't just to kill, but to kill without anyone ever proving it was murder,
then GWS, despite the technical hurdles in development and outcome prediction, becomes unparalleled. The potential for perfect deniability is its unique strategic advantage.
Okay, that makes sense. And it shifts us nicely into Section 5, talking about how these stealth attributes fit into the bigger picture of genetic weapon systems, GWS, and how they might actually be used strategically.
Right. GWS leverages these stealth characteristics for situations where political distance, plausible deniability is absolutely paramount. We need to think not just about the agent, but the deployment mechanism too, which has to be equally subtle.
And that subtle deployment often relies on covert delivery.
Yes. The sources often envision deployment through small-scale special operations, maybe even exploiting routine international travel or commerce, something that allows the perpetrator to deliver the agent without being detected during the act.
Avoiding immediate detection, avoiding immediate retaliation.
Exactly. And because the resulting outbreak looks so much like a natural event, maybe it's written off as a bad flu season or an unusual cluster of autoimmune cases, or a slight uptick in cancer rates, attributing that event back to a deliberate, sophisticated attack takes a very long time. Weeks, months, maybe even years, if ever.
Let's revisit those specific use cases where stealth is really the key asset. We touched on covert assassination.
Yeah, where that lack of attribution is the absolute top priority. You need to eliminate a high-value target, but maintain complete diplomatic deniability. A GWS design, perhaps for that rapid, untraceable cardiac failure or neurological collapse we discussed, it's uniquely suited.
Gives the perpetrator plausible deniability.
Maximum plausible deniability. It can act as a silent, invisible calling card. Even if intelligence agencies strongly suspect foul play, if the pathologist can't provide hard physical evidence of an external agent, then political action, sanctions, retaliation, it all becomes incredibly difficult.
And this loops back to those long-term cryptic effects, like the oncogenicity we discussed earlier. The attack doesn't have to be immediate. In fact, maybe delayed deterioration is sometimes preferred.
It could be, absolutely. Especially if deniability is the highest operational goal. Why risk a high-profile, sudden attack that draws immediate attention when you could potentially achieve similar political or strategic aims by causing physical or mental decline over time?
Through, say, cancers triggered by those oncogenic mechanisms or progressive neurological damage from engineered autoimmunity.
Right. You create instability through slow, untraceable disease progression. Think about the political impact of creating a decade-long public health crisis characterized by rising rates of chronic illness and unexplained mortality in a rival nation or specific group. The health crisis itself becomes the weapon, and attribution remains nearly impossible.
And the scope isn't just limited to human targets, right? This has implications for food security, global stability, too. Let's talk about non-human targeting.
This is another deeply concerning area, the potential misapplication of genomic information as an anti-plant or anti-livestock weapon. The key vulnerability here is the sheer uniformity of modern global agriculture.
We rely so heavily on these vast monoculture plots of staple crops, huge fields of corn, wheat, rice, where genetically the plants are almost identical.
Exactly. We've essentially created enormous genetically uniform populations, and that makes them incredibly vulnerable to a highly specific, tailored genetic pathogen.
Like an engineered fungus or virus.
Precisely. You could design a targeted fungal blight, maybe a viral agent, perhaps with elements of stealth or delayed impact. It wouldn't necessarily wipe out the crop overnight in a dramatic fashion.
It could be subtler.
Yeah, maybe it just reduces yield significantly over a season, or makes the grain have poor nutritional value, or causes it to spoil much faster in storage. Because the entire monoculture lacks the genetic diversity needed to naturally resist it, such a tailored pathogen could cause massive economic and social disruption.
Destabilizing a whole region without an obvious attack.
Right. The resulting food shortages, the economic chaos, maybe localized famine, that becomes the weapon. And it could all be traced back, initially at least, to what looks like just a natural blight or an unusually bad harvest or some stubborn, inexplicable crop failure.
This really broadens the scope of who needs to be thinking about biodefense. It's not just doctors and soldiers. It's agricultural health experts, economists, public health officials involved in food security. They need to expand their surveillance thinking radically.
Absolutely. The potential targets are much wider than just humans.
Okay, that brings us towards the end, Section 6. Let's try to synthesize all these capabilities and talk about the challenge this next generation of bioweapons poses specifically to the modern clinician, the people on the front lines.
Right. The core message from the sources is that the ability to manipulate organisms using tools like CRISPR, advanced synthetic biology, it fundamentally changes the game. The possession or even just the potential development of genetically engineered pathogens firmly places them in the category of next-generation biological weapons.
They're moving out of the purely theoretical realm.
And into the realm of plausible, immediate strategic threats. It's not science fiction anymore.
And what specifically defines these next-gen characteristics beyond the stealth and latency we've covered? How are they enhanced?
Well, the ability to modify organisms makes it possible to deliberately create or enhance pathogenic characteristics that specifically make things harder, clinically and for public health. The sources highlight 4 key areas.
Okay, what are they?
First, potentially increased survivability, meaning the agent can persist longer in the environment or maybe remain viable longer outside a host. Second, increased infectivity, making it easier to transmit between people.
More contagious.
Exactly. Third, increased virulence, making it cause more severe illness or have a higher fatality rate. And fourth, maybe the most critical point for the physician trying to treat a patient, increased drug resistance.
That resistance piece again. We talked about engineering it in from the start.
Right. And that capability fundamentally changes the prognosis for a patient. It means that even if we do manage to identify the pathogen, maybe through that deep sequencing we discussed, our standard treatment protocols might be completely useless right out of the gate.
So the patient is already critically ill, the clock is ticking faster than ever, and you're dealing with an infection that's resistant to your go-to therapies.
Precisely. These combined capabilities enhance survivability, infectivity, virulence, and multi-drug resistance, on top of the stealth mechanisms they demand that we really rethink our standard approaches. They require a level of vigilance in the medical community that goes, well, far beyond just routine infectious disease surveillance as we know it.
We need to be prepared for things that look weird, that don't follow the textbook.
Atypical clinical presentations, resistance patterns that defy expectations, and these delayed onset chronic illnesses that really challenge our basic understanding of cause and effect in medicine.
Okay, let's try to pull this all together then for everyone listening. We've really walked through 3 primary modes of stealth that directly challenge clinical practice. First, the genetic latency, the potential oncogenicity, the long delay.
Right. Second, the clinical mimicry using autoimmunity or those untraceable regulatory peptides to make the illness look perfectly natural even on autopsy.
And third, the technical evasion engineering the bug to simply bypass our standard rapid diagnostic tests.
And understanding these conceptual threats, even if they seem futuristic, it really underscores, I think, a professional responsibility. There's a need for radical vigilance in the clinical setting.
Especially when faced with the unexplained.
Exactly, especially when dealing with atypical pathology. That diagnostic line, the one separating a naturally occurring, maybe idiopathic disease from a deliberately engineered attack, that's becoming the critical frontier.
So when you, as a clinician, see those atypical presentations or maybe unexplained clusters of really severe autoimmune flares that aren't responding to standard therapy, or sudden deaths without a clear cause, especially in people who shouldn't be at high risk,
It has to trigger a higher level of scrutiny. And that scrutiny needs to go beyond just the standard infectious disease panels. It probably needs to start incorporating demands for more forensic-level pathological analysis, maybe even deep genomic surveillance in certain cases.
It almost sounds like you're saying the medical community, particularly pathologists, oncologists, rheumatologists, they need to start thinking a bit like counterintelligence analysts sometimes, connecting subtle clinical dots back to potential, maybe distant exposure events.
It's an uncomfortable thought, but perhaps necessary. We need to be open to possibilities that currently seem unlikely. And that leads us, I think, to our final provocative thought for you, the listener, to consider as you go back to your work.
Okay, let's hear it.
If a truly sophisticated, delayed onset biological attack can be made to appear completely indistinguishable from, say, a statistically significant but natural rise in certain cancer rates or severe autoimmune conditions, what kind of radical infrastructure changes would we need? Changes that go way beyond traditional infectious disease surveillance.
What would be needed to even begin the process of attribution in that scenario?
Exactly. Do we need some kind of dedicated, maybe global division of forensic epidemiology, one focused entirely on trying to link cryptic chronic diseases back to potential remote or past exposure events?
And thinking about that,
Yeah.
what are the ethical boundaries? If we start doing that level of deep genomic surveillance on populations to look for these engineered triggers, what does that do to our current ideas about medical privacy and consent?
Heavy questions. Definitely something to mull over.
Indeed. A lot to think about. Thank you for diving deep with us today on this complex and sobering topic.
Always a fascinating, if unsettling, discussion. Thank you.